This video is for the hydrogen peroxide lab. So first you'll need safety glasses. Once those are on, you're going to have one person at your table get the yeast. So you'll need a half a teaspoon of yeast in your first flask, like so. You'll also need 50 milliliters of warm water. I got this right from the sink. Doesn't need to be exact, about 50 is fine. And add that to the yeast, so that'll be the second person that gets the warm water. Give it a little swirl to wake up. So yeast is a living organism, it's a fungus, and just like when you're making bread, you usually have to activate your yeast in warm water. So we're going to let that sit to wake up. In the second flask, we're going to have our third person at the table measure out about 50 milliliters of hydrogen peroxide. Hydrogen peroxide, I got this from the grocery store. You can get it like CVS. So we normally use this to treat wounds, like first aid cuts, so it'll kill bacteria. And we're going to have the third person add it to the other flask. Giving our yeast a little bit more time to wake up. So when mitochondria are doing cellular respiration, the products would be water and carbon dioxide, but occasionally, instead of making water, H2O, they make hydrogen peroxide, H2O2. And that would be really bad to have inside cells. It would damage the inside of your cell. So we have, like all living things that have mitochondria and do cellular respiration, they have an enzyme called catalase inside their cells, and anytime that enzyme bumps into hydrogen peroxide, it does a chemical reaction to break it down. So what we're trying to find out in this lab today is what do they break it down into. Alright, so I think my yeast is warmed up, so I'm going to put both of these in my bucket. And I'm going to add the hydrogen peroxide to my yeast. So we're using yeast as a surrogate here. Since it's a living thing, it also does cellular respiration. And so it also has catalase enzyme. And it will also start a chemical reaction with that hydrogen peroxide. So you can see there's bubbles forming and they're rising out the top. So in order to figure out what is in the bubbles, what gas is in there that was made as a new substance, the best way to do that is to do a flame test. So if you think about hydrogen peroxide, the formula H2O2, you could predict a lot of different things as the products. So you might think it just breaks apart into hydrogen and oxygen gas, or it could break apart into water and oxygen gas, or maybe water and hydrogen gas. So we're not really going to be able to tell if it made water since we already had water in the flask. So we're really just going to be able to, we're going to have to just test the bubbles and see what we get. So if we get both hydrogen and oxygen, we'll know it's the first one, or the second one just oxygen, or the third hydrogen. Alright, so we're going to do a flame test. I'm going to take a splint and I'm going to light the end of it on fire and set our camera here. So if it were oxygen, we would expect the flame to just get brighter. If it was hydrogen, it would make a loud popping noise and it would be a big fireball. So I'm just going to step back and I'm just going to put the flame in the bubbles and we'll see what happens. if you're really good, you can get it to reignite too. I'm going to see if I can get it to reignite. Okay. I think we're done. Okay. So we're asking the question, did we just see the flame get brighter, which would tell us that it's oxygen being made along with water, or if we heard a really loud pop and the whole thing kind of explode into a fireball, and that would tell us it's hydrogen.